so today we're going to talk about genome editing, some of the different techniques in genome editing, and give you some examples and some awesome videos. All right, so what we mean by genome editing is the ability to go and change the sequences of the genome, right? That's what editing means. How we do this is we use programmable nucleases that enable targeted genetic modifications in culture cells as well as whole animals and plants. So as long as you can inject and transfer DNA, protein, and RNA into the germ cells, eggs, or embryos, then you can modify their genome. Okay, so that's the only thing you should be able to do. If you can do this in any organism, you could change it. Does that mean we can change human genome? Do we have the technical capabilities? Yes, right? We can inject into the embryos. We could transfer DNA, anything. So yes, humans are one of the organisms that you can change. Pretty much any organisms you can inject or transfer DNA, you can do this. So some of the organisms that people have done this already are mosquitoes, silkworm, pig, cow, primates, etc. So what are the different types of technologies that people use? One is called zinc finger nucleases. The second one uses transcription activator like effector nucleases called talons. And the third one, which is CRISPR and is the crispiest, is the RNA-guided engineered nucleases, RGNs derived from bacterial clustered regularly interspaced short palindromic repeat, so in short CRISPR, and Cas enzyme, which is named because it's CRISPR associated. All right, so what can you do with genome editing? You can do a whole bunch of stuff, including reverse genetics, high-throughput functional screens, live visualization of genomic loci, and you could do this. You could use this technology. It's kind of like a tool, and you're basically limited by your imagination. Possibilities are endless, right? And it also raises some ethical concerns, and because of its capabilities, it's been declared one of the most important breakthroughs in technology development in the past decade or so by scientific communities, and has made its way through scientific news literature, right? And so it was one of the world-changing ideas. And then when it makes to popular culture, then the reality gets a little distorted, and you get headlines like "custom babies not far off," right? So I think it's still far off, but yeah. So popular news don't capture the science as well as we hope that they could. Anyway, so let's go to the actual molecular biology of this thing. All right. So what do these technologies allow us to do? They allow us to target a specific site in the genome, and they allow us to target a double-strand break at the site that we targeted, right? And you learned in the repair lecture what double-strand breaks will do. The cells won't tolerate this double-strand break, and it's going to try and repair it, right? And in the repair of this break, if it uses the non-homologous end joining pathway, what you will have is you will have typically small deletions at the end of the DNA due to that blunting step, right? Or you can have insertions, which we didn't go through, but non-homologous end joining can also lead to small insertions. So you will have variable length indels, typically, sometimes very small insertions. You can also use the homologous recombination pathway, and in this case, instead of letting the cells to find a template to choose for homologous recombination, what you can do is you can provide the template, right, and force them to use this. What you can do is you can provide a donor template that has exactly the same sequences at these homology arms that are surrounding the double-strand break. And when the double-strand break site homologous recombination machinery finds this donor template, what it will do is it will copy this template and it will insert that into the genome. So you can use, change the sequences, so whatever sequence you want to insert, and put it in this donor template and surround it by the sequences that are homologous to the double strand break ends and have it inserted in your genome. Okay, so you can make precise insertions or modifications through homologous recombination, or you can make non-precise variable length deletion or insertions if you just let the cells do its own thing. All right, so we said you can make mutations or knockouts or replacements. Is this forward or reverse genetics? Reverse, right? Because we know what the sequence is, we target that sequence. So we could typically do these with species that we've already sequenced, or we know some sequence information. Okay? All right. So what are zinc finger nucleases? This is one of the programmable nucleases. They are programmable because what we can do is we can engineer protein sequences that are forming these zinc fingers that can bind and recognize sequences in the genome. Okay, so you can make these little zinc fingers that will recognize a specific sequence. So a zinc finger protein at the N-terminus contains a nuclease called FOC1. And FOC1 nuclease, when it homodimerizes, it becomes active. Okay, so what you do is you engineer zinc fingers that are going to bind to these sequences on the left and the right side of your target region, and you space these things a certain distance apart. So they're going to be about 5 to 7 base pairs apart, and that spacing is going to bring these two FOC1 proteins together so they can homodimerize, and they can now cut the DNA at this site. Okay. So each of these zinc fingers will recognize 3 to 5 3 base pairs DNA sequence, and you can change the protein sequence of each zinc finger to recognize a different set of 3 nucleotides. Okay? So what type of proteins contain zinc fingers? How did we get this information? Yeah, transcription factors that contain zinc finger DNA binding domains, right? By studying these zinc fingers, then researchers figured out which type of zinc fingers are capable of binding to which DNA sequences, right? And then you could engineer and use these zinc fingers to bind and recognize specific DNA sequences, right? And then you can add multiple zinc fingers together to bind longer and longer sequences. Does that make sense so far? Any questions? Yeah?
So like if the total sequence is about 6, or is about 11 to 13 base pairs, wouldn't there be like a lot of sites in the genome where you can find that sequence?
Exactly. So that's a very good concern, and I had that as a question later in the lecture. But yes, this is what we call off-target effects. Off-target means it can cut at regions that you didn't intend it to cut, right? So what the researchers do is when they're selecting the sequences, they do a little bioinformatics and make sure that that sequence is not present anywhere else in the genome. What that means is it's as much different as they can possibly make it from the other sites of the genome, right? So that's why knowing the genome sequence is important for these type of reverse genetics experiments. So you can make sure that you're targeting the specific site. How can you increase specificity other than making sure that you didn't hit a region of the genome? One of the things you can do is you can make it longer and longer sequences, right? Make it recognize longer and longer sequences, and that will make it more and more specific because the chances of finding that longer sequences is going to be lower, right? Any other questions? Very good question, yeah. All right. So what can you do with this kind of technology, right? So this was one of the first programmable nucleases that were produced. And one of the things you can immediately think of is, can we use this for gene therapy, right? And edit cells that you can actually deliver into the humans again. So as a proof of principle, what the researchers have done is they used zinc finger nucleases to create a 32 base pair deletion in the HIV co-receptor CCR5. So it was known before in previous researches that this deletion actually confers resistance to HIV injection, HIV infection, right? So what they did is they took some cells and they disrupted the CCR5, making this deletion using zinc finger nucleases. And then that resulted in about 50% of the alleles. So in a population of cells, they were capable of changing the sequence of the CCR5 in about 50% of the alleles, 50% of the chromosomes. And then what they did is they used this in a mouse model of HIV infection. And then they asked, are these CD4 plus T cells now resistant to HIV infection or not? And they have shown that, yes, they conferred some resistance, right? So what that means is this. You have an organism which is normally susceptible to HIV infection. You can take out its cells, engineer it the way that you make it resistant to infection, put it back into the organism and make that organism more resistant to HIV infection, right? So they have shown by proof of principle that this is possible. This is something that you can do. All right. So the second type of programmable nucleases are called TALENs, transcription activator-like effector nucleases. Again, they're using the knowledge of transcription factors and how they bind to specific DNA sequences and harness that power to engineer a protein and target it to a specific site, right? In this case, these are different type of protein domains, so they're not zinc fingers, but each of these transcription activator-like effector has a tandem array of 33 to 35 amino acid repeats, and each of these recognize a single base pair in the major group. Okay, so you can engineer these repeats to recognize any sequence that you want. So the target sequences of TALEN pairs are about 30 to 40 base pair in length. Again, what they're doing is they're fusing this DNA targeting domain, right? So this DNA binding domain that they engineered using TAL sequences and fuse it to the FOC1 enzyme and create a specific cut site here. So by changing the protein sequences that can that will specifically recognize specific sequences in the genome, they can target any sequence in the genome, right? And again, the effect is exactly the same. You create a double strand break. Yes?
So based on that, TALEN sequence is where the FOC1 will cut?
Yes, exactly. So you have to make sure that your TALEN sequences are a specific distance apart so that FOC1 can homodimerize and cut. If you put it too far apart, it won't be able to cut. If you put it too close together, it won't be able to cut. So it has to be a certain distance. Right. Any questions? All right. So that's exactly the same thing, and people have used TALENs to edit the genome as well. Now we're going to talk about CRISPR-Cas9. So in the CRISPR-Cas9, the recognition of the DNA is different. Now, what we are not using transcription factor-like domains to bind and recognize sequences. What we're using is an RNA, and it's complementary to DNA to target a nuclease, okay? In this case, what we have is we have the CRISPR-associated nuclease called Cas9, okay? It's shown in this light blue kind of structure here. And this protein complexes with a couple of RNAs. One is called the tracer RNA, so this is a transactivating RNA. And this is a structural RNA that has some stems and loops. And this complexes with the guide RNA, okay? So this is the guide RNA here. And the guide RNA sits in the protein in a way such that 20 base pairs of this guide sequence will make interactions, complementary interactions with the target DNA. Okay? So that's going to be the complementary strand. And target this whole complex to the DNA. And this is called a protosphere adjacent motif called NGG. When the protein finds its good target sequence that the RNA is complementary to and recognizes this PAM sequence, it's going to cut the DNA here. All right? So in this way, what we have is we have a RNA that's associated with a protein, and the RNA sequence is going to find the complementary site in the DNA and target that Cas9 and nuclease to that specific site. All right. So that's how it normally exists. But people can engineer such that the guide RNA is a single RNA now, and this actually just is basically it helps the researchers to synthesize a single RNA to do what they want. So that's just a technical way of making things a little bit easier for molecular biologists. So we call this, which is if you work in labs nowadays, this is what you're going to talk about a lot, which is the single guide RNA, which is a fused RNA that takes the tracer RNA and fuses to the guide RNA, which contains the guide sequence. So you can make this complex Cas9 with the single guide RNA, and it's going to work just as well as the separate. Okay. All right. So what does it do? Once the Cas9 with the single guide RNA finds its complementary site in the genome, it's going to cut it right upstream of the PAM site. Okay? And the PAM site is NGG. N means any nucleotide, followed by GG. That's what the sequence that this Cas9 needs. All right. So here I had asked, genome editing can have an off-target effect. That means cut a place where you didn't want to target. So we talked about why, because the complementarity is for a short sequence. So if by mistake you had that sequence somewhere else in the genome, or a sequence that resembles very much this 20 base pair guide sequence here, but might have one or two mismatches. Sometimes the Cas9 sgRNA complex makes a mistake, and it recognizes even that sequence and it cuts it. Okay, so giving you off-target effect. Why is it important that you do not have an off-target effect? What if you were targeting a specific gene, but you had off-target and you accidentally cut in a few other places in the genome? What might happen? Yeah?
Large deletions due to homologous.
You could have mutations, right? You could have large deletions, you could have small deletions, you could have chromosomal translocations, because, like we said, cells do not tolerate double-strand breaks. It's going to try and find a way to make sure that double-strand break is gone. And the way it will repair is non-homologous end joining or homologous recombination, and it can make mistakes. Yeah?
When it recognizes that PAM sequence, does it not cut right at the end of the guide DNA? It cuts 3 before?
Yeah, it cuts at a specific distance from the PAM. Yeah. All right. So how does this look structurally, right? So this is a very hot area of research. People really want to understand how the CRISPR-Cas9 works, and there has been a lot of structural experiments. So people have crystallized the Cas9 with the target DNA sequence and with the CRISPR RNA shown here in red and the DNA shown here in yellow, and figured out what domains of the Cas9 protein are capable of cutting the DNA. So these are the nuclease domains, and found out which amino acids in the nuclease domains are important for this cutting, and what is the structural basis of that recognition, etc. Right? So here's the the Cas9 enzyme. Each of the domains are shown in different colors, and the single guide RNA and the DNA are shown. So what the researchers have done is they used atomic force microscopy to figure out how the complex sits on it so that they can watch this thing live, okay? So before I show that movie, so they're using a Cas9 from Cas9 that first basically recognizes the protospacer adjacent motif PAM. And then the Cas9 RNA initiates unwinding of the DNA at the PAM proximal region, because that's the region it's going to search for homology, right? So it recognizes the PAM, opens up the DNA, so it's going to see if the guide RNA is complementary or not, right? It's searching. All right, so, and it's going to create an R loop. What that means is that it opens up the two DNA strands and inserts the guide RNA in there, okay? So creating that R loop, which is two DNA strands open with the RNA. And if this RNA is complementary to the DNA, it's going to create an RNA-DNA hybrid, right? So it's going to anneal there. And it's going to displace completely the non-target strand. After that R loop is formed with the RNA-DNA hybrid, that activates the Cas9 enzyme to cut the target and the non-target DNA strand respectively, okay? So the target strand is complementary to the RNA guide, and the non-target strand is the other strand that was displaced. Okay, how does this actually look? So here's a movie. Let's see if I can play this. Yeah. So you're seeing one of the domains that keep moving in the Cas9, and the movement of that domain is associated with when the Cas9 enzyme is going to cut, and that you see that little part that got kind of left out, that's the DNA that was cut, right? So magenta is showing that domain that's going to be cut, and again you're going to see the DNA that's being cut and released. And come on, there you go, right? And here's another movie. You have the Cas9 right on the DNA. It's amazing. You're seeing the DNA, guys. That strand is DNA, right? And Cas9 is cutting the DNA right in front of your eyes, right? And this is what CRISPR-Cas9 works, right? So it kind of stays attached to the other strand for a little bit, and then it falls, okay? Any questions? All right. So how did the researchers actually come to finding this mechanism of genome editing? This was done through a lot of basic research over many years, okay? And there are a couple of characters here that were very influential in finding this mechanism and actually characterizing the Cas9 enzyme. This is Emmanuelle Charpentier and Jennifer Doudna, two researchers that actually collaborated to figure out how this thing works, okay? And they weren't actually out there looking for enzymes to cut the genome. They kind of stumbled upon it by studying a very curious biological question. And that should tell you that sometimes going after very biological curious questions that might not necessarily be immediately translational, you can actually find very important clues to health and other things. All right? So what they were studying is bacterial immune system. So in the bacterial immune system, in bacteria and archaea, these RNA-guided DNA cleavage systems provide adaptive immunity against invading phages or plasmids. Okay? So what happens is that when a viral DNA invades the cell contains the Cas9 complex, which cleaves the DNA and takes up a small portion of that viral DNA, a little portion, which we call the spacer, okay? And then inserts that into a clustered repeats, okay, that puts that spacer in. And each of these spacer here, these are little short DNA sequences, are coming from different viruses, okay? It's kind of cataloging oh, okay, this virus entered, and here's a little sequence that corresponds to that virus, etc., etc., right? Cataloging all the viruses that has been coming here. And the same locus, which is the CRISPR locus, and there are different types. This is the type 2 locus, encodes for the tracer RNA, the Cas9 enzyme, several other genes that are important for this initial recognition and cleavage of the viral DNA, as well as these spacer DNAs that are separated by these other sequences. All right, so what do they do next? Next time they get invaded, right? So that is that they learned it, right? They learned the DNA sequence of the invading virus. Next time they're invaded, what happens is that viral DNA, the Cas9, the tracer RNA, will also complex with this guide RNA, and the guide RNA is being transcribed from these spacers, okay? So this guide RNA and the tracer RNA is going to complex with the Cas9 enzyme. And when this complex here, so this is the little blue that's coming from the guide RNA from this spacer, right? This spacer sequence has a complementary site in the viral DNA. The Cas9 enzyme is going to cleave it such that the viral DNA is no longer capable of infecting the cell, right? Yeah.
How does the cell survive the initial infection?
The infections are not perfect, right? So if it was perfect, it would have 100% death, right? But most of the viruses that can't do that, right? So some of them will always survive. Any other questions? All right. So same guide sequence is actually over here, right? But why doesn't it cut its own DNA? The reason why it doesn't cut its own DNA is it requires a certain conformation of the RNA. Okay, the RNA has to be such that the guide RNA should recognize and be complementary to its target, and not complementary at the 3' and some on the 5' end, right? So this conformation is required for targeting of the non-self DNA, targeting of that viral DNA. In its own genome, the sequences are such that at the 3' right here and the 5', they are complementary to these repeat sequences that are surrounding each spacer. And when the guide RNA is complementary to its own genome like this, the Cas9 enzyme is inactive, so it's incapable of cutting the self DNA. Right? So there is a structural basis for recognizing self from non-self DNA. Bacteria have other types of adaptive immune systems, right? So, for example, how do bacteria protect its own DNA from restriction enzyme cutting? It's a very similar idea, right? Bacteria uses restriction enzymes to cut invading DNA that are coming from the viruses and phages and stuff like that, right? So, but it doesn't, these restriction enzymes doesn't cut its own DNA. Why? What can you think of? What might the bacteria be doing to prevent this? You probably learned this in POB, I'm pretty sure. What kind of modifications do bacteria to their genome, to their DNA? Humans, vertebrates do it too. What about methylation? DNA methylation, right? So what bacteria does is it methylates its own DNA so that the restriction enzymes, even though the same sequence, target sequence are in the bacterial genome, because of the methylation, either it can't bind or it can't cut. Okay? So that's how it protects itself. And in humans, we also methylate, right? But for other reasons. All right, so let's now talk about some of the things you can do using genome editing. And we briefly talked about reverse genetics. We're going to talk about some systems people have designed to target, activate genes or repress genes. And you can use these type of things in high throughput sequencing. You can use this technology for live visualization and some ethical concerns as well. Okay, so we just talked about how you can actually use this for reverse genetics. So you can use, you can create short deletions or insertions using non-homologous end-joining repair of the break, or homologous recombination if you provide a template. So you can engineer one or multiple cuts in the genome, right? So if you wanted to create a large deletion, you're not going to be able to accomplish this with one cut, right? Because that's going to go through the non-homologous end-joining pathway, and it's going to create small deletions. If you want to delete a whole big region, a whole gene, create a knockout, what you can do is you can actually cut the genome in two sites, right? So this is the region that you want to take out, for example, and you can let the cells repair by ligating this end of the DNA to that end of the DNA, essentially removing this using non-homologous end joining. Okay? Or you can make sure that this is going to happen by providing a DNA template. And if you want to insert certain sequences, for example, you want to knock out a gene and insert a selection marker, right? So that you can select only the cells that have this insertion. What you can do is you can clone your selection marker so that you can make a DNA fragment that contains your selection marker, and at the end of that DNA, you provide sequences that are exactly the same to the two ends of the DNA cuts that you've created here, right? And then you can insert your selection marker here. You went through this type of technology with Professor Siegel a lot, so I'm not going to go through this, okay? So you can insert your selection marker in. The reason why you might want to insert selection marker is when you make these double strand breaks, the cells can choose several paths to repair it, right? And then you need to screen those cells to find out the ones that repaired the way that you wanted it repaired, okay? Does that make sense? Any questions here? All right. So we do this in our lab a lot. A lot of molecular biology labs do this because it's a great tool, it's easy to use, and we can manipulate the genome to understand genetics of whatever process that we're working on, right? So in a couple lectures before, we have learned about this dosage compensation complex in C. elegans, right? It looked like this. And we said that we could find a DNA sequence motif, right, that was present at the binding sites of this dosage compensation complex, right? But we found this thing, but we didn't know if it is actually important for this complex to bind to those sites. And what do? Like a good geneticist, there are two things you can do. There's the first thing that you would do is knock out the sequence and find out what happens, right? So that's what we wanted to do here. So this is a binding site. So this is a ChIP-seq experiment, and we're looking at the binding site of one of the proteins in the dosage compensation complex. And you can see these very nice two binding peaks, right? Two sites that it's binding. And each of these sites contain this motif here. And we wanted to knock out that motif. So what we did is we targeted that motif with a CRISPR. Okay? So those two binding sites are under two repeats. This is repeat 1 and repeat 2. And we designed CRISPR guide RNA to these pink arrows shown here. Okay? And the PAM sites are shown in this gray color. And we also designed a set of PCR primers shown here in pink. And each of these PCR primers have a name, C65 forward and reverse, etc., etc. Okay? So when we target these sites using the CRISPR Cas9, it made a double strand break right here, and it was right around the motif. And we were hoping to get a mutant which has a deletion that was deleting that motif out. Okay? So then what we did is we injected this into the gonads of hermaphrodite, this C. elegans worms, and then let them make progeny and screen their progeny by PCR to see which one of these worms now contain this deletion. And we're doing that by doing a PCR using these two primers. Okay? So here's the result of that experiment. So we're screening 6 progeny from that injection. This is the wild-type length of the DNA when you use the PCR in wild-type animals without any CRISPR-Cas9 injection. Okay? And then here are the DNA lengths that we get by PCR from each of our progeny. Okay? So by just looking at the results of this gel, which one do you think is the mutant line? 6, right? Because the product that you get out of the PCR from the 6th progeny number 6 is a little shorter. And what kind of mutation do you expect here? A short deletion, right? So we got the PCR product to shift down a little bit. So it's a little bit shorter than the wild type. All right. So what happened, right? Did we actually now all of a sudden not see any binding? That's exactly what we saw. So this is the ChIP-seq data from wild-type cells, wild-type animals, right? And you can see these 2 binding sites. And when you when you use the progeny number 6, where this motif is completely eliminated, what we saw is that the binding was also gone. Okay? So using this experiment, we show that this DNA sequence is important for the binding of the complex to that site. All right. Any questions? Okay. So now that we have a tool in which we can target a protein to a specific site in the genome for cutting, we can actually engineer that protein such that we can remove its enzymatic activity, right, and just use it as a tool to target specific sites. And how do we do this? So researchers have crystallized the Cas9 protein with the DNA and the RNA, and they determined which are the enzymatic pockets that are going to cut the DNA, right, and which are the amino acids that are critical for this. And then what they did is they mutated these amino acids. So histidine 840, that's the amino acid at the 840th location. When they change it to an alanine, and at another site, this amino acid to an alanine, what they could do is they could completely remove the enzymatic activity of the Cas9. So they call this dead Cas9, which is awesome, right? Dead Cas9, because it's catalytically dead, right? So it's dCas9. So if you use then dCas9 with your sgRNA here, what you can do is you can target your protein to any specific site in the genome, and your protein will bind. And it turns out dCas9 is amazing. Unlike a lot of proteins that fall on and off, they have a rate of a very high dissociation rate. dCas9 is very stable. Once it goes and finds the spot, it just sits there. Okay? So now you have a tool where you can target whatever you want at a specific site in the genome. What do you do with this, right? What do you do with that kind of tool? You can do several things, and I'm going to show you 2, in which you can actually regulate the transcription of a specific gene. Yes?
Is there a way to make it fall off if you wanted to?
Probably, and that would be a protein engineering problem, right? Now you would try out different mutants based on the structural properties and see if you can actually control on and off dynamics if you want to. All right, so what can you do? What researchers have done is they developed a CRISPR-mediated transcription regulation system. So what you can do is you can take your Cas9 and clone it such that you can fuse a protein, whatever protein you want, to the C-terminus of Cas9. Okay? So you fuse whatever protein you want to the Cas9, and then you can use an sgRNA that's gene-specific, and then target your protein here, right? Whatever protein you want, to a specific site in the genome. And the way that you can do this is you can transform the construct that is encoding for this dCas9 fused to your protein of interest and a construct encoding for this sgRNA, transfect these into cells, and the cells will express this. And so what you're going to have is that your sgRNA, guide RNA, that's going to be complexed with your dCas9 that's fused to your effector protein, and then you can change this effector protein to whatever you want and bring that. So one of the things that you can do is you can fuse a transcription repressor domain or activation domain, right? And target your dead Cas9 to the promoter of a gene to turn off or on or off that gene. Does that make sense? Okay, so let's look at an example. So here's the proof of principle. What they're doing is they're creating 3 constructs. Okay, one construct is expressing only dCas9 on itself. Okay, so it has a promoter that is pretty highly expressed, and that downstream of that promoter you clone Cas9, and you can fuse it to some fluorescent protein so you can actually see it. All right. In another construct, what they've done is they have dCas9 with the fluorescent marker, and then they fused a repressor domain from a protein called CRAB. So they fused the CRAB repressor domain to dCas9, and this construct, when is encoding, when it's in the cells and gets transcribed and translated, it's going to produce a dCas9 protein that's fused to fluorescent and the CRAB repressor domain. And then they create a third plasmid, right, a third construct that is going to be encoding for the single guide RNA. So when it's transcribed, it's going to make that single guide RNA, which is going to complex with dCas9. All right. Then so they chose this CRAB repressor to reduce transcription. So how do these repressor proteins work in eukaryotes for repressing transcription? What do they do to repress transcription? Repressor domains. RPD3? Okay, what in eukaryotes? How the repressors work? Come on, guys, we had midterm 2, and the average was pretty good. Very good, right? So these repressor domains can recruit co-repressor complexes such as the histone deacetylase complex that's going to deacetylate things and keeps things down, right? So it's going to shut off transcription, right? So that's what the repressor domain is. And so they're going to take that repressor domain and fuse it to this dCas9. Normally, the repressor domains are going to be targeted to a specific site in the genome because they're part of transcription factors that contain DNA binding domains, right? The repressor proteins have DNA binding domains and a repressor domain, and that's how they're targeting different sites in the genome. This time, we're taking the DNA binding domain, switch it with the dCas9. Now we can take that repressor domain and put it anywhere else in the genome, right? Okay, so then you inject these things, and then what you need to do is you need to have a target site, and in this case, the researcher chose a reporter construct, okay? They want to see if they can target and so that they can change the expression of this gene called EGFP, which encodes for a variant of the GFP protein, so they can measure the amount of expression of this gene using the GFP signal, okay? All right, and so the target site is right here in the promoter. So what they're doing is they're taking this sgRNA construct and choosing either dCas9 alone or dCas9 with the CRAB repressor domain, and taking the reporter construct, co-transfecting into cells, and then measuring the GFP fluorescence to see what happens to expression. All right. So here's the data. So what we have here is that if you transfect all your dCas9 and sgRNA, but don't put any reporter into the cells, you're not going to have any GFP expression, okay? And that's what the gray line is here. And what we're having here is a histogram. So this is the fluorescence measure of the EGFP. So the higher the fluorescence, the more expression of that EGFP is happening, okay? And the counts are coming from each of the cells. So a distribution like this showing that most of the cells in your dish is not expressing EGFP. So all you see is this gray line is the background signal coming from whatever the autofluorescence of the cells are, right? So there's not much signal at all. So then if you put the reporter construct, but you put the dCas9, but you don't provide any guide RNA, right? So that the Cas9 is not going to be able to bind to the guide RNA. Then what you see high expression from the construct because you have a strong promoter here on its own is supporting the high expression of the EGFP, and that's what you see in the black line here, right? So you have higher fluorescence signal when you have the reporter. Then what you do is you can put the reporter dCas9 fused to the Crab repressor domain and the sgRNA. Now what's happening is that this sgRNA is going to target the dCas9 Crab repressor to the promoter, and what you see is that now in the presence of the reporter construct, you have very little EGFP expression, right? Equal to almost not having this EGFP construct at all. So you completely repress the transcription of this, okay? And here's one more experiment. What they've done is you only target the dCas9 without the repressor domain, and with the sgRNA, what you see is a reduction in the fluorescence, showing that the expression of the gene has been repressed to some extent, but not as much as when you had the Crab repressor domain. Okay? So some repression was achieved without the repressor domain. How do you think this happened? When you target the dCas9 to the promoter, right, without the repressor domain, you were capable of reducing the transcription of GFP. So this is the expressed, right? You're reducing to about here on average, right? That's how much repression you get with dCas9 alone, sitting at the promoter, right? So why do you get that repression? Yes?
Well, because if it has really stable binding, it's going to block other things from binding.
Very good.
That have less stable binding.
Excellent. So if it is stably binding the promoter, by physically, it's going to prevent other things like initiation factors coming and binding. What type of mechanism does this remind you of?
Repressors and prokaryotes.
Very good. Sigma factor, how the lac repressor, by sitting at the operator, prevents the sigma factor coming and binding, right? And then, but the repression is not perfect. So it can still, the initiation factors can still come in and repress a little bit, but when you add the repressor domain, which is going to bring the core repressor and histone deacetylation, etc., you reduce the amount of repression from this promoter a lot more, right? So you increase the repression by adding that repressor domain. Excellent. Any questions? All right. So this was to repress a gene, right? So if you wanted to repress a specific target gene, this is what you would do. You can also use exact same strategy to activate a gene. What do you need to change? Instead of repressing, what do you need to do? Yes? Yeah, instead of fusing a repressor domain, you're going to fuse an activator domain, right? That's essentially it. So this is the experiment, how they've done this. So what they did is they have the dCas9, and they have fused it into a activator domain, in this case coming from a protein called VP64. Again, they have the fluorescent protein. Then they have another construct making the sgRNA, and then they have a reporter in which they are expressing GFP as a reporter, and they have several target sites at the promoter region of this GFP. And this time they're using a promoter that's weak, right? On its own, it shouldn't express very much. Only if you put a lot of activators, then it's going to be turned on and start expressing. Okay? So that's the difference in the two reporter construct. In the repressor construct, we had a strong promoter, right? Could support a lot of transcription, so we can measure the repression. In this case, we are using a weak promoter, so there's not much transcription on its own. We're going to see how much the activator can activate. All right. So we're going to co-infect these things and then find out what happens. So in this case, the data is represented as a bar plot. So we're looking at the fluorescence of the GFP reporter here on the y-axis. So the higher the fluorescence, the more expression of this reporter, right? So when you put the dCas9 with the activator domain, and here it's shown when it's minus, it's not transfected. When it's plus, it's transfected. So when you put the reporter on its own in the cells, this is how much background fluorescence you get. Okay, so that's however much this weak promoter can support expression. That's the background that you get. When you put the dCas9 on its own without the sgRNA, okay, so when you do this, so if you put the sgRNA by itself with the reporter, you don't get much. When you put the dCas9 on itself, in its own, without the sgRNA, you don't get much expression at all. But when you put both of them together, in which the sgRNA is going to complex with dCas9 and target it to the promoter of the reporter gene, then you can achieve 25-fold activation of that promoter, right? So by bringing dCas9 fused to an activator domain, you can activate that promoter. So how does that activation occur in eukaryotes? What is the activation domain doing? Eukaryotic transcription initiation, regulation, mastery topic. It's going to bring transcriptional coactivators such as SAGA, right? And it's going to help TF2D, etc., etc. Okay, good. What else can you do? You can do a lot of things. You guys can think about a lot of things, but one of the things that people have done is you can visualize individual loci in the cell. And this was very, very difficult to do live, okay? You can do this with not live cells. You can use techniques to visualize a specific genomic locus in the cell. What technique can you use? This was in the midterm. Fish, right? DNA fish, you can visualize. But you can do this only after you fix the cells, okay? So you can't do this live. But sometimes you want to know the dynamics of how different genomic regions move around in the cells. We know that's important for a lot of processes, right? So you want to create a technique to do this. So what they've done is that they fused a fluorescent protein, EGFP, to the dCas9, okay? And then they used a bunch of different sgRNAs to target this dCas9 to a specific locus in the genome. And then by using fluorescent microscopy, you can see these little dots here, right? They can see that those are the sites in the genome where the sgRNA is targeting the dCas9. Now, in live cells, they can track these dots, see where they're going, to be able to measure the different, that specific genomic site's location in the cell, which is, yeah, so this is pretty cool. All right, so these are some of the techniques that you can do. You can come up with a lot of other things. It's basically CRISPR has a way to target specific sites. All right. You can also take CRISPR editing and apply it, right? So one of the things the researchers have done is use this in different animals and show that it works. So one of the first things that some of the researchers have done is they used it in monkeys. So the experiment is basically you have your target sgRNA and your protein, and you can inject it into into the embryos after fertilization, and then you can put that into surrogate mothers and then find which of your progeny actually has the genome edits that you actually look for, right? So in this experiment, the scientists targeted 3 genes simultaneously in more than 180 single-cell monkey embryos. So they're doing in vitro fertilization and then injecting the eggs that are targeting 3 different genes, right? And so the 83 embryos actually survived this injection. And so they put these embryos in surrogate mothers, which resulted in 10 pregnancies. So it's not a very efficient, you can see that it's not too efficient, right? Starting from 180, we went to 83 and then 10 pregnancies. And one of them led to a birth of a pair of monkeys with mutations in the 2 of the targets, PPAR gamma, which helps regulate metabolism, and RAG1, which is involved in healthy immune function. So they were able to change the genome of the progeny. Right? If you can do this in monkeys and primates, you can do it in humans, right? And that was actually also done in this experiment where Junhil Huang and the colleagues at Sun University said, okay, can we do this in human embryos, right? And so what they targeted here is one gene called HBB, which is the human beta globin protein. So there's a lot of ethical issues, right? So if you start with embryos that you're going to, you know that can survive, you're basically editing a human, right? So to circumvent that, what they did is they used embryos that have chromosome copy number problems, right? In the previous lecture, we talked about how in humans, we have a lot of embryos. When people get pregnant, a lot of the embryos that have chromosome copy number differences, that is missing a chromosome or having an extra chromosome, those embryos don't survive. They get miscarried after a few weeks, okay? So what they did is they chose those embryos, those with copy number variation, the chromosome copy number problems, which we call aneuploidy, missing or having extra chromosomes. It's called aneuploidy. So they used some non-viable embryos, aneuploid embryos. They injected 86 embryos and waited 2 days. And of the 71 embryos that survived, they were able to test 54 of them. And 28 of them had genomes that were successfully cut, but only a fraction of those contained the replacement DNA. So they were trying to change the DNA sequence of this gene by using homologous recombination and providing a template, right? So that would be gene therapy kind of thing. If there's a mutation, you want to correct it. You don't want to just cut it, right? Cut the gene. So a very inefficient process, but it's still doable, right? So this is very inefficient. So less than a few percentage of the embryos actually contain the changes that you wanted to do. So the researcher said that if you want to do this in normal embryos, you need to be close to 100%. So you need to have 100% efficiency, so you don't actually kill the embryo, right? And you don't want any off targets. So basically, his team also said that there was a surprising number of off-target mutations that they assumed to be introduced by the CRISPR-Cas9 acting at the other regions of the genome, right? So now researchers are trying to get this off-target effects much lower, increase the efficiency of homologous recombination, etc. But there's also a big debate in the field, like, is this ethical to do, right? And that's what that designer baby news article coming after this report, right? So it's possible, but just because it's possible, we don't do a lot of things, right? I could just jump off the cliff, but I prefer not to, right? It's just because it's possible, sometimes it's not a good idea. All right, so where else can you use gene editing? One of this application is called gene drive, okay? So what you can do is you can target entire populations. So far, we always talked about targeting just one organism, right? By injecting its embryo, etc., etc. But in gene drives, you can target entire populations. And the way that you can do this is you basically stimulate bias inheritance of a particular gene to alter the allelic frequency in an entire population. So it can change wild populations, and some of the applications would be changing the mosquitoes so they can't carry the malaria parasite, right? So that would be one application for this, or dengue fever, etc. So the idea is this. So you introduce a mosquito into the population that encodes at a given locus for a gene that cuts the DNA, right? So this could be any endonuclease gene. In this case, it could be a CRISPR-Cas9 construct that you inserted here. So when it mates with a wild-type fly, what happens in the progeny of these mating, heterozygous progenies, meiosis, the gene-drive chromosome can cut the wild-type chromosome, right? So you make sure that your endonuclease is targeting the site where you inserted your gene-drive allele of the endonuclease. When it cuts here, because the surrounding regions here and here contain homology to these sites, it can repair this break by homologous recombination, copying the sequences from the gene-drive chromosome into the wild-type chromosome. So some of the progeny will contain, again, two gene-drive alleles. That means exactly what you put here in the nuclease. Some of the progeny is going to repair this using non-homologous recombination, because you can't control this process, okay? And these guys are going to have just a mutation in the target site. And some of the progeny might not have any cut, right? And they're not going to have any repair, right? So, but this process is going to repeat itself over and over again. And what will happen is that the frequency of the mosquitoes containing the allele that you introduce is going to increase with time, okay? Because you cheated Mendelian inheritance and you changed. So over time, the frequency of your allele is going to increase. And this is actually an idea that's hotly debated. People think about applying this, and the biggest target are mosquitoes because they're pretty bad, right? They carry a lot of bad diseases. But there are also problems in changing whole populations in the world, right? Okay, so what are some of the medical applications of this? There are very interesting medical applications. One of this is organ transplants. So in humans, some of our organs in terms of size and function are very similar to pigs, right? There has been a lot of talk about trying to get pig hearts being transplanted into humans because they're about the same size, etc. But one of the problems with pigs is compatibility, right? The human body is going to think that it's not the right thing because it's not the right thing. And the other problem, one of the other problems, is that the pig genomes contain a lot of retrotransposons that are still active. So if you actually introduce the pig cells into human cells, etc., if these retroviruses jump, then they can jump all over the place and then mutate our genome, right? So one of the things that the researchers have done is, can we actually inactivate all of these retroviruses using CRISPR? And they were capable of doing this. And so then putting the first step towards trying to humanize these pigs so we can actually use their organs, right? All right, if that disturbed you a little bit, there's some more undisturbing ways of using this CRISPR in medicine. And so one of the most exciting ways to use CRISPR would be combining it with stem cell technology, right? So how do you do this? So the idea is this. So you have a patient, right, and it has some problems with its organs or whatever process, immunity, etc., etc. Because it has mutations, right? Mutations give us a lot of these diseases. What you can do is you can isolate some skin cells from this patient or other cells and create induced pluripotent stem cells, right? So you take them back into their differentiation and make induced pluripotent stem cells. Then you can put these in a dish and do CRISPR and screen for the right clone that has your corrected mutation or whatever, right? Whatever change you want to make this in the genome of that. And then you can grow more of it. Then you can redifferentiate to whatever cell type that you want to replace in the human, right? One of the ideas is neurons, but of course this is a very difficult thing to do. And then if you have a way to transplant back to the patients these corrected cells, then you can cure the patient, right? So this is the dream of the medical fields these days, to combine CRISPR with stem cells. And it's not just a dream, it's actually also already started, and there is already a clinical trial that is started. And in this case, they're starting with a cell type that's a little bit easier to handle, so because you can inject these T cells, right? So immune cells. So what they've done is, they removed T cells from 18 patients with several types of cancers. And the idea is to use cancer immunotherapy. So what they're trying to do is engineer these T cells such that they can attack and reduce the cancer cells in the patients, right? So they're removing these T cells from 18 patients and performing 3 CRISPR edits. These edits, they have to very carefully choose based on a lot of basic research that was done for 20, 30 years, okay? So this is accumulated knowledge that they're using. So what they're doing is they're inserting in the genome of these cells a gene that's encoding for a receptor for NY-ESO1, which is a protein that's often present on certain tumors but not on healthy cells. So they're directing the T cells to find the cancer cells because cancer cells specifically express that protein. So they're making a receptor for that protein, okay? The second thing, they disrupt the gene encoding PD-1, which sits on the surface of the T cells and help dampen the activity of the cells. So what happens is that tumor cells, they hide from the immune system of the patients. And the way that they hide is they activate the expression of PD-1 in the T cells normally, and that reduces T cells activity. So the cancer cells trick them, okay? So this genome edit, what it does is it prevents that. So these cancer cells won't be able to trick these engineered T cells, all right? And then they also do one more change in the T cells. It's a primary receptor so that the T cells are more active, okay? So this is all years of immunology research in cancers, figuring out what is the best method. And then what they've done is then the researchers then infuse these edited T cells back into the patients and see what's going to happen to the cancer cells, right? And so hopefully, if everything goes well, these T cells will attack the cancer cells, they will leave the healthy cells alone, right? And it will help these patients to recover. Yeah?
18 patients seems like a pretty small study size. Is that because the risks are high?
There's a lot of ethical things to go on here, right? Because they don't know what's going to happen. So the choosing of the patients is one thing that limits, right? And you don't want to start out big, right? So in clinical trials, there's phase 1, phase 2, phase 3, etc. This is the first step. Let's see if it's going to work, right? With 18, they can get a foot, like understand what are the problems, what they can change, and it's a small number enough where the committees felt reasonably good about trying and not going just right away, right? But this is ongoing, so we don't know what the result is. Any questions? All right. Happy Thanksgiving, and we'll see you in a couple of weeks. I mean, I'll see you in a couple of weeks. You will see Professor Siegel soon.